Okay, what we have to do for topic 4.4.4 is describe the application of DNA profiling to determine paternity and also in forensic investigation. Now, some of you are so excited about crime scene investigation that you definitely want to do something like that. All right, so this is the stuff that you'll be doing. All right. So we can collect DNA sample from blood, saliva, or semen and amplified using PCR. No, those are not semen that you actually, on a Navy ship, not that semen. All right, satellite DNA non-coding is cut with specific restriction enzymes to genetic fragments. I got it. Okay. Not like Navy sailors. Okay. All right, individuals. Gotta mix it up a little bit, folks. A little fun. Allie's all business. She's always all business. All right. So you're gonna have that individuals with a unique fragment length due to variable lengths of their short tandem repeaters, STRs. You're gonna hear about STRs quite a bit. Apparently, I got farther in fourth grade than I did with you guys. So the smaller the fragments are, the quicker they're going to move through the gel. So they're going to go to the bottom of the gel. All right, DNA profile can be analyzed according to need. And two applications, we have paternity testing. If you want to be on the Maury show. Ever watch Maury? He's not the father, my neighbor's the father. Come on in here. I'm going to test the neighbor. No, not father. You are the father of this child. All right. All right. Forensic investigation. So identifying suspects and victims. All right. Next. Okay. Paternity testing. Paternity testing. Children inherit half of their alleles from each parent and thus should possess a combination of their parents' alleles. Forensic investigation, suspect DNA. Should be complete match for the sample taken from a crime scene that conviction is to occur. What is happening now at a rapid rate, at an exponential rate? People that got accused of killing somebody in the 70s, 60s, 70s, early 80s are getting off because their DNA doesn't match the actual crime scene. So it's a ton of people getting off. All right, so we take a look. Did a little example here. Mother, child, male 1 and male 2. We don't know which male is going to be the dad. So we go on the Maury show. I know she's a cheetah. I know she's a cheetah. I don't know which one is my baby's dad.
Baby dad.
Baby dad. All right. So, with that being said, so you have to get tested. And then Maury pulls out of his envelope, and the results are in. And this is what happens. So we got the child. Here's the child's DNA. It needs to be a mix of mother and either one of these two. So if you look at the child, we got the blue from dad, and we got the pink from mom. So we have a nice mix. This guy's off the hook. Breathes a sigh of relief. And the same thing with a crime scene. So we have the DNA from a crime scene, and we do all the sequences of suspects and the victim. We find that the suspect matches exactly with the crime scene. This is not an exact match, paternity testing, but crime scene is obviously an exact match. All right, so let's do this. All right, so here we go. On your activity sheet. In paternity test, DNA samples from 2 possible fathers. It's on the back. It's on the back. See, it's on the back. See, all the way on the back of your activity sheets. Activity sheets. All right, paternity test. All right, in a paternity test, DNA samples from 2 possible fathers and from the mother are compared for the offspring's DNA. In this procedure, the samples are digested with a type of enzyme that cuts DNA at specific sequences. what do we call those enzymes? What are those enzymes called? Say it. Nah, well, the specific sequences are genes, but what is the enzyme? What's the term for it? A restriction enzyme. All right, so yes, number 1 is on the board. Hopefully you're answering number 1. All right, and what do you need to add? Number 2 is added there. Number 2, I just said what the answer to number 2 was. There you go. Might be a couple other things, but restriction enzymes. What is compared in the paternity test? DNA samples from 2 possible daddies. 2 possible daddies. One's a sugar daddy, one is not. One has caramel and one doesn't. Thank you. Thank you. Thank you. Got nothing. Got nothing. Okay. All right, so we're going to add the enzyme. Look at Pac-Man going to work. What's Pac-Man doing? You missed it. You missed it. Let me go back. All right, the DNA is loaded. All right, so we're going to load this on here. Pull out the little thing. It's loaded on a gel according to size. It's separated according to size by gel electrophoresis. There you go. All right. How is the DNA separated? According to size. According to size. All right, so now we haven't determined dad yet. We haven't determined dad yet. Okay? Now I know if I was still teaching at Brunswick, this might be real case. That would never happen here. None of you people would ever be like that, right? Right? None of you would ever be in this situation. Yeah, okay, that's right. All right. All right. Every band of the offspring's DNA must match a band in at least one of its parents. Let's first consider the matches with the mothers. 3 bands match the mother. All right, 3 bands match the mother. See if you can see the 3 bands. Let me hear. Here, I'll have somebody come up. All right, Margaret. Come on up here. See if you can circle which ones match. Okay. Ooh. Okay. Very good. Very good. All right. You came up to Gasland. All right. Oh, she did it right. The 3 remaining bands must be shared with the father. I'll go find out if you baby daddy. Go. Only one possible father shares the 3 bands of the offspring, therefore. This must be the father. Oh, all right. Very good. So here's what we got. We got Darian or Valerie. And the winner is Darian. Congrats. Congrats. Are you excited?
Yeah.
Yes.
No, Darian, he's not excited. He's ready.
Uncle Darian.
Uncle Darian.
He's excited, guys. Give him a break. He's excited. Yes. They have to match the remaining bands that did not come from mom. The remaining bands that did not come from mom. Unfortunately, Valerie, you are not the father.
Shocker.
Well, you never know. Sometimes you gotta run the test. Life will find a way. Life will always find a way. What movie?
Yeah, that movie.
There's a huge
That definitely.
There's like the next two slides.
Oh, yeah.
Okay, now, the next two slides, you look at the other handout I gave with you because I had my cartoon on the top. Look at the handout, right in your notebook, right there, Jenny. I see
I
it.
see it.
All right.
That's all you gave us this one too.
All right. That's how I roll. All right, the Human Genome Project was an international cooperative venture established to sequence a 3 billion base pair around 25,000 genes in the human genome. Okay, now, for some reason, Bill Clinton said that it was a tie between the private and public world. Everybody knows who Bill Clinton is, or president, former president. Said it was a tie, but that he is actually incorrect because Celera Genomics did complete the human genome first. The possible outcomes of this project include mapping. We know the number, location, and basic sequence of the human genes. Screening allows the production of specific genes. Probes to detect. It's right here on this paper. Look, see that paper you got last class? You got that? Yeah, look at that! Whoa! All right, screening. Allow for the production of specific genes and probes to detect sufferers and carriers of genetic disease conditions. And medicine, genomic medicines, pharmacogenetics, and rational drug design. Ancestry will give us improved insight into our origins, evolution, and history and migratory patterns of humans in mitochondrial DNA. All right, we began to sequence the genomes of non-human organisms, and we have in vitro hybridization. So now you already saw my wonderful joke.
I don't think God never said that's part of the joke.
Okay, way to kill a moment. All right. 4.4.7. State that. Oh, I love to state that. Those are my favorite. When genes are transferred between species. What?
So can you do it?
Let's see. I'll try it. I'm going to try it. All right, let me see. When genes are transferred between species, the amino acid sequence of the polypeptides translated from them is unchanged because the genetic code is universal. What does that mean? What does that mean? AUG is what? Do you know? Start in U. What about cow? What about in a snail? Yes. What about in a piece of grass? How many amino acids are there? 20. How many amino acids are there possible in a snail? How many are there in grass? How many are there in buffalo chip? You know what a buffalo chip is? Yeah. What? Oops, that's right. All right. Okay, buffalo chips. If you ever say, if anybody ever offers you like cow chips or buffalo chips or dog chips, just avoid them. Trust me. All right. This means that the genetic information from one organism could be translated by another. It is theoretically transferable. Current examples of transgenic modification: corn, sweet corn, potatoes, papaya. You know what papaya is? What is papaya? Huh? It's a fruit. Oh, it's my favorite. Meat, dairy products. 22% of all dairy products are injected with HGH, human growth hormone. How about honey, cotton? Estimates of about 30,000 different products. Take a look at these mice. No, they're not as creepy as you really think they are. Those are glowing eyes and glowing tails that they transmutate into from a glowing fish. Pretty sick. A fish that glow, they took the genes out, put them in these mice.
Can they do that to humans?
Yes.
Wait, are they connected?
I want that.
They're not connected, no.
So are we Yeah, could they give me gills?
What do you think that benefit is? They glow in the dark?
They cross the human and they glow. So you can see them at night, and then if you
It doesn't, in light, it looks normal, like a normal mouse. Take out the ears, right?
Is the one in the middle like the father?
That's they put a normal one in the picture and the two genetically modified ones on the outside.
So.
It doesn't. This animal doesn't know any different. There's no. In the day, it looks exactly the same as any other mouse. It's just at night, under a black light, it glows.
Just gonna eat like crazy.
Correct. All right, 4.4.8. Outline a basic technique used for gene transfer involving plasmids, a host cell, bacterium or yeast, restriction enzymes, endonucleases, and DNA ligase. This is how they did it. This is how they are going to fix you from your future ailments. By using a little bacteria, changing the DNA, and then injecting it into you, and then getting into your DNA and changing your DNA so you are not sick anymore. All right, so what happens is we remove a plasmid. Does anybody remember what a plasmid is? Circular DNA that can exist and replicate by itself. A gene of interest is removed from an organism's genome using a restriction enzyme, endonuclease, which is cut a specific sequence. So a gene, so it's cut off. So we're going to try to, we want whatever we want in our future thing, whatever trait we're trying to get. And it's amplified using PCR technology. So we've got to get billions of copies. All right. Note the sticky ends of unattached hydrogen bonds. All right. So we'll have sticky ends where that DNA polymerase is going to go across for your PCR. All right. So the restriction enzyme is going to cut that thing out, and then we're going to replicate from there. And we have two sticky ends. They're going to be on opposite sides of each other. Okay. Now let's take a look. Step 2, digestion and ligation. All right. The plasmid is cut with the same restriction enzyme that used to excise the gene of interest, to pull away that gene. Cutting with certain restriction enzymes may generate short sequences. Sequence overhangs the sticky end that allow two DNAs to construct together. And the gene of interest and plasmid are spliced together by DNA ligase, creating a recombinant plasmid. Recombinant plasmid is what you're going to then, well, I'll tell you what we're going to do here after this. Interesting. Transfection and expression. The recombinant plasmid is inserted into the desired host cells. This is called transfection for eukaryotic cells and transformation for prokaryotic cells. Underline transformation and transfection so you know the difference. The transgenic cells will hopefully produce the desired trait by encoding by the gene of interest expression. You want pink eyes, you want green eyes, you want whatever eyes. This is a process you can use to get those. Really.
Oh, purple.
Not really used at that level yet for humans, but obviously we just saw it work in monkeys. So that means it works in humans so far. All right. This product may need to be subsequently isolated from the host and purified in order to generate a sufficient yield.
What if it gave you black eye?
Well, that would suck if you had black eyes. All right. So this is what it looks like. So if I'm a hemophiliac, what's a hemophiliac?
Humans cannot clot them.
Can't clot their own blood from, say, just keep bleeding. So if we're going to treat a hemophiliac via the isolation of the human factor 9 clotting protein from transgenic sheep milk. Yeah? Okay? So we're going to have our human. We have our human cell. And then we find the human factor 9 gene, the clotting factor. All right? So we're lacking in that. We don't have that. So we take a bacteria cell. We pull out the plasmid, which is right here, the DNA. We take this, cut out a part of the plasmid, doing what? Using what? What do you cut out with? So let's see. Let me just write it down here. You need this. All right. Can't clot blood. All right. Human cell. We know this is the nucleus, or whatever. And then we have the bacterial cell. What is this little part right there? What's that? Plasmid. This is the nucleoid region. So what did we do right here to the plasmid? We cut it with what?
Lasers.
We didn't use lasers. What did we cut it with?
The same restriction enzyme that was used to.
All right, restriction enzyme. All right, so we cut out the restriction enzyme. We took what we needed here and we inserted it into the plasmid and we got recombinant, which is a combination of what you want and the bacterial. Combination of desired outcome. And original plasmid plus original plasmid. Okay. There you go. Then we have a sheep egg. So what do you think we're going to do with the sheep egg? I'm going to show you an actually done under a microscope. But we're going to take that. We're going to pull out. We're going to take this recombinant DNA. We're going to insert it into the sheep egg. We have a transgenic sheep. And then from here, we have the human factor 9 from the sheep's milk. And hopefully, we can then get that into the human. And then be good to go. And use that clotting factor. And if somebody has issues with hemophilia, well, they're going to get injected with this factor so they don't have it. In order to treat hemophiliacs who can have blood, we use a normal person's human cell and process the gene transfer. Please take any notes. Okay, I already did that. Sorry. Just for other teachers that might not go over it. I don't know. Okay, 4.4.9. State 2 examples of current uses of genetically modified crops or animals. I'm going to give you 2 crops, 2 crops and 3 genetically modified animals. So there's 2 on this page and then 1 other one. Tomatoes. The flavor saver tomato. You ever hear of that? It's in the grocery store. Have been engineered to have an extended keeping quality by switching off the gene for ripening and thus delaying the natural process of softening of fruit. So they can stay in your grocery store longer.
Are these more expensive?
Yes, no. You don't even know. They're just the tomatoes you buy. Engineering of crops provide protection from insects. Maize crops, BT corn, have been engineered to be toxic to corn borer by introducing a toxin gene from a bacterium, Bacillus thuringiensis. All right, and then we have the frankenfish. Anybody hear of the frankenfish? Genetically modified salmon to provide more food. The frankenfish, named by opponents, or aqua advantage salmon, contains a growth hormone borrowed from the Chinook salmon and a genetic switch from the ocean that activates the antifreeze gene. There's a normal salmon in the bottom. There's the frankenfish behind it. A little bit bigger? So what do you think that fish is going to probably do? Eat more of everything else? Become more of a predator? Maybe have a bigger impact on the environment they may think. But it's okay because you can still eat. All right, and then a couple animals I have talked about here. Engineering animals to enhance production. Sheep produce more wool when engineered with the gene for the enzyme responsible for production of cysteine, the main amino acid for the aid in the acid in the keratin protein of wool. And the other one I talked about was sheep engineered to produce human alpha-1 antitrypsin in their milk can be used to help treat individuals suffering from emphysema. What's emphysema? What's that? Yeah, breathing. You have trouble in your lungs, you can't breathe as well, get air, so most of the time you're on oxygen or so forth. Okay. Could probably hit up one more here. I think this is it, right? This is it? Are we almost done? All right, so I didn't, what?
Yeah, this.
Almost done. Okay. Example. I would like to finish them up so we can get ahead and then make everybody jealous. All right, maize. Introducing the bacterial gene encoding a toxin to the European corn borer. What benefits do we have? Discuss potential benefits and potential harmful effects of an example of genetic modification. Here it is. Here was a benefit of making this corn unbreakable by the corn borer. Bore means make a hole in. Allows for the introduction of a characteristic that wasn't present within the gene pool. Selective breeding could not have produced desired phenotype. So no matter how much you wanted to make something that was resistant to the corn borer, you couldn't do it by breeding different strains of the corn. Results in increased productivity of food. Production requires less land for comparable yield. That's good. There's some right in Frederick. We have the same thing in Frederick, just a bunch of farms with it. Less use of chemical pesticides, thus it's not getting into your drinking water, reducing the economic cost of farming. That's good. Cannot grow in regions that previously may have not have been viable. So we don't have to deforest things. We can grow it maybe in the desert. We could grow it in not rainy places or super rainy places. What could be harmful about it? Could have a currently unknown harmful effects. Toxin may cause an allergic reaction in a percentage of the population. Don't know. A lot of this stuff we don't know. We just do it, and we make more food and then we eat it. Nobody has any idea. Or you're like, you look like you're 18 when you're 12. I mean, maybe that's another harmful side effect. Or 25 when you're 12. Accidental release of transgenic organism into the environment will result in competition with native plant species. Cross-pollination, introduced from weeds, weed growth, reduces genetic variation and biodiversity. Corn borer may play a crucial role in the local ecosystem. So you might disrupt the whole ecosystem. Like I said, the frankenfish, how big that fish was, it may have an issue of eating everything else, and thus the smaller species can't have anything to eat. Clone. All right, define a clone. A clone is a group of genetically identical organisms or a group of cells derived from a single parent. So I wanted to put this little picture up there. Okay. Okay, well, I tried. All right, outline a technique for cloning using differentiated animal cells. All right. Somatic cell nuclear transfer. I'm going to show you a video on how this works. Is a method of reproductive cloning using differentiated animal cells. Differentiated animal cells are the ones that, where we started making the certain lines that we wanted. Who can remember what the undifferentiated cells are? What do we call those? Stem cells. So we're beyond that point. They've already differentiated. A female animal is treated with hormones such as FSH to stimulate the development of eggs. FSH is follicle stimulating hormone. That's what makes women have their periods. All right. The nucleus from an egg cell is removed. Enucleated. Enucleated means to remove a nucleus. Thereby removing the genetic information from the cell. The egg is fused with the nucleus from a somatic cell of another sheep, making the egg cell diploid. And then what they do is they shock it electrically. It's delivered to stimulate the egg to divide, and once the process is begun, the egg is implanted into the uterus of the surrogate. And let me show you what it looks like. Oocyte enucleation. Who is smart enough to tell me what this means? Oocyte enucleation at this point. All right, so egg, nucleus removed. You can sound really smart, or you can just say egg nucleus removed. You can say oocyte enucleation. All right, developing embryo will have the same genetic material as the sheep. This is Dolly. This is how Dolly was made. All right, check it out. Here we go. All right, so what is cloning? There you go. There's the first two human clones right there. What?
What year they cloned?
Whenever they were born. Not really a clone. All right, so check it out. Let me pop this up. This is an egg. Let me turn it down. The lights up. All right, see the egg? That's an actual egg. Here comes the doctor.
Oocyte enucleation is the removal of the oocyte's endogenous chromosomes using a DNA stain to first localize the genetic material, and it lasts by death for its removal.
Okay, so the egg is out. They pulled what from the egg? Nucleus. Pretty sick, huh? It's ridiculous. Now, let's do the cell transfer.
Cell transfer involves placing a cell from the animal we are cloning next to the oocyte side effects.
This is crazy.
Who's those two again?
I mean, doesn't that make you think of what this guy's doing on a video on just some website?
Oocyte enucleation is the removal of the oocyte's endogenous chromosomes using a DNA stain to first localize the genetic material and a glass pipette for its removal.
That's a glass pipette. Does that make you feel uncomfortable?
Cell transfer involves placing
Yeah.
a cell from the animal we are cloning.
It's happening
next
every
to
day.
the oocyte side effects.
Thousands of times a day. Okay, bang. Went in the same hole he plucked earlier. Now, whatever he wants this to be. So, what would be different if this was, we've done it in sheep and other animals, but why not, think about what they could do with that. I'm not going to go any further with that. All right, so.
Maybe like